Hi, night class. Miss Celeste here. I'm going to give you your lecture on hematology. Starts on page 1158 in your big book. If you have any questions, you can always let us know if I didn't explain anything good enough, but I am going to try to make this pretty quick for you since it's recorded video instead of live teaching. All right, hematology, the study of blood. The MA's role. If you worked in a lab, you would be collecting and processing, maybe storing these samples. So it's important that you know your normal values and understand your test results so that we can educate the patients. Very important for patient education, of course, and recognizing critical situations. So if we have an emergency, if we have levels that are so abnormal it's emergent, we need to be able to catch that for the physician and be able to let them know. Okay, we already know all about OSHA and CDC guidelines. I think we'll jump on to blood function, formation, and components. All right, so I want you to know the functions of the blood. What they do are transportation. So they transport everything in your blood, your O2, your nutrients, everything that we need to nourish the body, and of course take away the waste, which is your CO2. So waste products and elimination, taking that out. It's also responsible for regulating your body temp, which is also of important function. Right, plasma, the liquid component, so like the water of the blood is your plasma. 90% of plasma is water, so that's a lot. A big percent of your plasma is water, and only about 10 is solid substances. All right, on to the next page. The formed elements of the blood, which are your cellular components. So under that, the formation of blood cells is hematopoiesis. Hematopoiesis. And we have red cells, white cells, and platelets. So red blood cells, white blood cells, and platelets, these are your formed elements of the blood. Important to know your formed elements of the blood. Red blood cells are called erythrocytes, which we already know. They transport O2 and CO2. And white blood cells are leukocytes and therefore defense. So they have to do with your immune system, and there's different types of white blood cells. I want you to know granular leukocytes. We have neutrophils, eosinophils, and basophils, and we have non-granular leukocytes, lymphocytes, and monocytes. So I want you to know all your different types of white blood cells, and it goes further deeper into that on in box 47.1. Box 47.1, it explains those a little better, and I'm not going to go too deep into them here, but if you want to read box 47.1 on your own time. All right, let's see. Number 3, last formed element of the blood. Thrombocytes or platelets are the same thing. I want you to know they're the same thing, have to do with clotting. So your platelets or your thrombocytes are really important for blood clotting, which can be very fatal if we have something wrong with that. All right, CBC stands for complete blood count. One of the most common tests routinely ordered in the office, CBC. You'll hear that and see that a lot. Under red blood cells and red blood cell tests. These erythrocytes carry your hemoglobin throughout the body, formed in the bone marrow and replaced every few months. Red blood cells are replaced every few months. Important to know that. We're going to talk about hemoglobin now in the second column. All right, so hemoglobin has two functions, so carrying O2 from the lungs and CO2 from the lungs. So important to know, when hemoglobin is carrying oxygen, it's called oxyhemoglobin, and when it's carrying CO2, it's carboxyhemoglobin. Important to know that. All right, under erythrocyte or red blood cell count. So the number of RBCs, important to know for adult male, is 4.5 to 6 million. 4.5 4.5 to 6. Important to know for adult male, 4.5 to 6. Normal female range is 4 to 5.5 million. 4 to 5.5 for females. Normal red blood cell count. Anemia, I think we've all heard of that, right? Anemia, condition where the blood has lower than normal red blood cells. So they have a low amount of red blood cells, and this can cause a lot of symptoms throughout the body. People with anemia are generally going to be pretty fatigued and maybe just not feeling up to par all the time. Let's see, on to the next page. Let's see, so the opposite red blood of anemia, is polycythemia, so where the blood has higher than normal level of red blood cells. Overproduction by the kidneys or overactive bone marrow, high altitudes, exercise, smoking, tumors, renal disorders, dehydration. These are all things that can cause that. So opposite of low, we have too much. All right, hemoglobin. On to hemoglobin. So your red blood cells contain hemoglobin, and hemoglobin consists of iron and protein. I want you to know one molecule of hemoglobin contains 4 atoms of iron. One molecule of hemoglobin contains 4 atoms of iron. Hemoglobin is responsible for carrying O2 throughout the body, and low hemoglobin may indicate iron deficiency anemia. So if we see a low hemoglobin reading, it would be important to report that to the physician. They can have anemia. This could be very bad. Normal values. Adult females, important to know, 12 to 16. So normal hemoglobin, 12 to 16. And adult males, 14 to 18. 14 to 18. So we're going to use an automated blood analyzer to test hemoglobin. It's a pretty easy test, just a quick capillary stick. We're going to use just a little bit of blood for a hemoglobin test. So just a little blood analyzer, and we can do that. That's in our scope of practice. On to the hematocrit on the next page. So the hematocrit, this evaluates the percent of packed red blood cells in the total volume of blood. And it's going to be collected in a tube. So you perform your phlebotomy procedure, you have your tube of blood, and then you're going to put it in the centrifuge. So there is a picture in here of the centrifuge, and you might have seen it in the lab before. It's a little machine. You put the vials of blood in it, and it spins it down. It's going to spin the tube for about 15 minutes at like a high speed. And that gives you your separated components. So the red blood cells sink to the bottom, the plasma floats to the top, and then the buffy coat, or the thin layer in the middle, is your white blood cells in the middle. And there is a good picture of that, figure 47.6 on page 1164. A really good picture, shows you the plasma at the top, which is green, the buffy coat in the middle, which is small, and then the red blood cells at the bottom. So good to look at that picture. That will give you a visual. A low hematocrit. So if it's a low hematocrit, it may mean anemia or hemorrhage. So it would be important, if you see a low hematocrit, to point that out to the physician. All right, the microhematocrit is similar to the hematocrit. It's just going to be small quantity of blood in a capillary tube. It's just same process, just a little amount of blood in a little tube. On page 1165 at the top, it shows you a picture of the microhematocrit. All right, on to erythrocyte indices. So red blood cell indices means the exact same thing. These differentiate the type of anemia present by indicating the size of the red blood cells and the concentration. So we're further testing to see more about this anemia. What type is it? What's it caused by? Okay, ESR on the next page stands for erythrocyte sedimentation rate, ESR. So also called the SED rate, evaluates red blood cells. It determines the rate they settle to the bottom of the tube, so it's like timed. They're going to time how long it takes for the red blood cells to settle to the bottom of the tube. I do want you to know we use the Wintrobe method. the Wintrobe method is how we get your ESR. In the second column now, so